Welcome to the epigenetic part of the lecture again. So last lecture we had a visiting or a guest lecture who discussed DNA methylation, but I want to give a very quick introduction of epigenetics. So people were started, or at least a lot of people were interested in epigenetics because they see these heritable changes in gene function that occur without the change in the DNA sequence. For example, from one cell to the daughter cell after differentiation, a blood lineage cell will know that it's already blood lineage instead of being a brain or a muscle cell. How does the cell remember this, right? If it's all encoded in the DNA sequence. And also when people are doing ChIP-seq for the transcription factor in different tissues, such as androgen receptor binding, say in prostate versus other liver or other tissues, you would see that even though transcription factor motif influence the transcription factor binding in the different cells, the factor will bind to different locations, and not all the motif occurrences were bound by the factor. And also, even with the transcription factor binding, it doesn't necessarily regulate the nearby gene. Sometimes it just parks there without influencing the nearby gene. And so what influences these? And that's kind of how a lot of people are interested in epigenetics. If we only look at the DNA sequences, kind of pretty much all the cells and also all the individuals have the same DNA, but this is actually packaged in different levels of the chromatin. The very basic unit of the chromatin is called a nucleosome. Basically, there are 8 histone proteins that's kind of forming a nucleosome, and then there's 146 nucleotide that's DNA wrapped around this 8 histone proteins in 2 circles. And so you can see here, the blue here are 8 histone proteins, and the red is the DNA wrapped around in 2 circles and then move on. And then there's another nucleosome and another nucleosome. And this can then be stacked up together to form this 30 nanometer fiber. And then if you zoom out even further, you'll see something like this. This can be even further packaged. You can imagine this as a rubber band. You keep twisting, and then some regions become really tight, whereas other regions are still pretty relaxed. During the metaphase of the cell cycle, the chromosome really gets very compacted, and at that time, no gene is transcribed. The transcription activities are completely silenced. But during the regular cell cycle, in the non-division phases, the cell will be kind of having more relaxed chromatin, and the really open chromatin regions will be active for transcriptional activity. And so understanding the epigenetic of the cell will tell us which locations are more compact versus more open, which will dictate what kind of genes are going to be transcribed. And so in the last lecture, we discussed DNA methylation, which are modifications on the DNA directly, but you can see there are different levels of epigenetics, and the very first level is nucleosome, and the positioning of the nucleosome also matters. So this is another view of nucleosome. You can see here this DNA double helix, and then it wraps around these 8 different histones in 2 circles, and then it will move on and then wrap around another 8 histones. And the basic unit of the chromatin is this nucleosome, which is 8 histones and 146 base pair of DNA. The position of the nucleosome is important in that very often transcription factors would prefer to bind to the nucleosome-free regions like this region, whereas if the region is occupied or the DNA is occupied by the histones to form a nucleosome, many transcription factors would not be able to come and bind to these regions. And so in order to really map the nucleosomes on the chromatin, there are experiments that can be done. We digest the chromatin with an enzyme called microcalcanuclease, or MNase. And this MNase basically will cut, it will have an easier time to cut in these open regions, nucleosome-free regions. And then you will be able to get just the nucleosomal DNA. And then you isolate the DNA and sequence them out very often now from the two ends, which the nucleosome is right in the middle, and the two ends are the two ends here and there, right? So using this approach, we can sequence out the nucleosome and see where they are. This is an example from a 2009 review. At that time, most people only sequence one end of the nucleosome. At that time, paired end sequencing wasn't so popular yet. But again, this is similar to like a transcription factor binding. You will see two peaks in here, which usually is to the left and the right of this nucleosome. And if you shift the sequence to the middle, that will give you a peak shape. And here we know roughly how much to shift because the nucleosome is 146, so you only shift about half of this, 73 nucleotides. Then you will see the nucleosome positions in the genome. In the reality is that if you can imagine, usually this experiment is done in millions of cells, and in the different cells, the nucleosome, it's like a pearl on a necklace or beads on a string. They can slide around. And so when you sequence this, you don't always see a lot of peaks because when the nucleosome are not well positioned, the nucleosome can kind of freely move around across the genome. And so the signal is kind of more even. But occasionally, you would see a very well-positioned nucleosome, which means across millions of cells or many, many cells, this nucleosome is always at this position. And then you will see a very well, peak kind of situation. And so there are different factors that influence nucleosome positioning. The first is DNA sequence. This people call it like a cis element or DNA influence, which is like this. If you look across the genome for AA versus TT or TA sequence, and then compare it to the locations where you have either CC, GG, or CG sequence in the genome, across across eukaryote genomes, you would see something like this, starting from yeast, then many animal species here will have this 10 base pair periodicity, which is the percentage of AATTTA fractions. It's a very small fluctuation, but it's kind of consistent across the a genome. If you just look at any region, you will see this kind of weak 10 base pair periodicity. This is because the DNA that's wrapped around the nucleosome, the DNA have a self-rotation in that double helix. You can see here, as the DNA wrap around the 8 histone, the double strand helix also have the self twisting in here. And interestingly, the GC sequence actually prefer to interact with the histone proteins inside, whereas the AT sequence does not like to face the histone proteins. And so if there are this 10 base pair periodicity, what happens is that usually the AT will always be facing outside, whereas the GC will be facing inside of the nucleosome. And because the double helix is a 10 base has a 10 base pair, each round of the double helix, the self twisting is 10 pair of nucleotides stacking on top of each other. And so basically the 10 base pair enrichment of the AT sequence will make sure that or at least will favor an AT sequence facing outside and GC sequencing face inside. And this phenomenon is only observed in eukaryotes when there are nucleosomes. For example, in bacteria and virus, there is no nucleosome, there's no histones, and therefore you don't observe this type of 10 base pair periodicity. And so you can imagine when there is the DNA that can either be here or here, right there, like within the 10 base pair region, there is a favored position when the nucleosome can fit snugly when the GC are aligned inside. But this actually have a very small influence. The bigger influence is really transcription factor binding. So as we mentioned, if you sequence nucleosome across the genome, in many locations, what you will see is like this. The nucleosome are not very well positioned. However, in some regions, in this case, you will see a very strong nucleosome-free region. The reason there's no nucleosome here is because the polymerase is a very strong transcription factor, and it recruits many other transcription factors and cofactors to bind to this region, and this will really squeeze out the transcription, squeeze out the nucleosome. And this region suddenly has no nucleosome, and because of this, the adjacent pearls on this necklace will also be kind of slightly better positioned. And then the next one will be better positioned. But gradually, as you go inside the genes, eventually, this is like it's like a track field. Everybody start from here, and then initially they are still synced, but gradually they will become out of sync. And you can see here, you lose the positioning of the nucleosome. And so you can see here, nucleosome positioning can influence transcription factor binding, but they are also influenced by transcription factors. For polymerase 2, which has a very strong propensity to bind there, they can actually squeeze out the nucleosome and force the nearby nucleosome to be better positioned as well, although this can gradually be out of sync over distance. And so this actually are translational nucleosome positioning, and they really are the more important factors influencing nucleosome positioning. And also these type of nucleosome positioning are better at predicting where transcription factor can bind. Okay, so that's the first part.